Hi, everyone. I'm Lisa Carnell. As Amanda said, I'm the program scientist for translational research in the Biological and Physical Sciences Division out of NASA headquarters. We are really excited about this opportunity and appreciate you guys, diving in and being sort of the first group to pilot this for us at the university professor level. And we're looking forward to the outcome of this and learning from you on how we can do better. And we really hope that this helps to grow this research area within your field and your university. So welcome, and we would love to hear any feedback you have as the course goes along or even following. Thank you.
Great, thank you so much, Lisa. And next, I'd like to introduce David Smith, who's currently the Acting Chief of Space Biosciences Division. So David, if you want to say a few words.
Yeah, welcome everyone. We're going to have an amazing couple weeks with you. As Lisa noted, we need you. We need you not only to provide feedback on how this pilot experience goes, but we need you in space biosciences and at NASA. And most of your amazing instructors are sitting within the division here in Northern California, the Space Biosciences Division. We'd love to have you come see our facilities sometimes if you're in the area, or if you love the biosciences that you get a taste of in the next couple of weeks and want an internship or maybe a postdoc or maybe a career opportunity. There's a place for you. We really need you. You're going to be on the cutting edge with some incredible instructors in the next couple of weeks, and we hope that that really lights a spark for you, whether you're a faculty member and get to teach this stuff or whether you're a student and want to pursue a career in biosciences. So have a great time. Thanks for your participation, and thanks to your instructors. Enjoy. I'll be popping in and out, and I'll put my email address in the chat box if you want to reach out and give us some feedback. Thanks.
Thanks so much, David. I really appreciate it. So next, I want to introduce you guys to Parag Vijayamampayan, the NASA Ames Space Biology Portfolio Scientist. So Parag, if you want to say a few words.
Thank you, Amanda. Good morning to all of you. I'm so thrilled and so happy to be here. This is our fourth HPCU workshop series, which Elven and myself, we have put together. So I'm really happy. Even after leaving JPL, I'm still working with my JPL colleagues, and we are working with you. So first of all, congratulations to all the educators and students who are participating in this amazing workshop. I am highly confident that you will take a lot with you, and the whole idea that you will not only take something with you, but you will give that back to your institutions and to your students, and it will be a self-replicating model for years to come. I just would like to thank all my JPL colleagues for putting this together and working with Ames. I cannot thank enough to Amanda and Lauren, who has doing all the heavy lifting here. But I'm so happy to see some of my other colleagues like Vijay, Kanisha, and I think Rachel is here, and it's fantastic to be here. And so welcome you all, and I will be gone from June 5th, but I would take the feedback from all of you, and I'll be in touch with Amanda. Thank you so much, and welcome again.
Okay, thanks, Parag. So next, I'd like to introduce you guys to Sylvan Costas, who is the Space Biosciences Research Branch Chief, also the Project Manager for Open Science for Space Biology, including GeneLab and Alsda, and is the lead of the Radiation Biophysics Laboratory. So Sylvan, if you want to say a few words.
Yes, can you hear me, guys?
Yep, we can hear you.
Okay. Well, yeah, it's been it's really exciting today. This class, this GeneLab for University for instructors and teachers. So we've been working on this for many years. I'm the project manager for GeneLab. The vision we had with GeneLab for University started quite a few years ago, and it's really moving forward now. And so you're gonna hear kind of the latest technology in RNA sequencing and other type of omics that we've done at GeneLab, but you're gonna have it in the context of space biology. And so the class is going to really have really two different aspects. One that's going to be quite novel to you and quite exciting, I think. And like you heard from David Smith, we really hope that you're going to take it to your institution and be able to teach to others. So that's kind of the idea, is we pass along what we know to others, and we grow this community. We're very eager to see more people doing omics on space biology. It's an exciting field. And anyway, I won't talk any further. Thank you for joining us. I hope you enjoyed the instruction, and we're looking forward to your feedback.
Thanks so much, Sylvain. Next, I want to introduce you guys to Sam Gabri, who is GeneLab's Deputy Project Manager. Sam, if you want to turn your camera on, introduce yourself and say a few words.
Sure. Thank you, Amanda. So I think all our managers did a great job introducing you and welcoming you. So welcome, and hope you guys enjoy this next week and a half. And I do want to say thank you for Lisa and Parag for really helping us push this along and setting up collaborations with JPL in order to do this year. So that was really great. And as David said, we're happy to share our emails if you guys have any questions. This is an opportunity to also network with us at NASA. So, please feel free to do that. And lastly, you guys are in great hands. Amanda and Lauren are great instructors. They know this subject really well and really enjoy doing this. So I hope you guys enjoy your time with us, and hope, just feel free to reach out to us after. And if you have any questions about GeneLab, and continue to use the data, because there's a lot there. So enjoy your week. And if you guys have any questions, feel free to send me an email.
Great. Thanks so much, Sam. Next, I want to introduce you guys to Elven Smith, who most of you already know is the manager for JPL's Planetary Protection Center for Excellence. So Elven, if you want to turn on your camera, say for viewers, and I know that you have a few slides that you want to share.
Yes, I'm muted. Okay. Thank you so much, Amanda. This has been wonderful. And yes, I did want to share a couple of slides. As Parag mentioned, this is our 4th year. We're super excited for the continuing funding that we've received from JPL to continue to put this forward through the ESD HBCU MSI Award funding. It's been an exciting time. And since 2018, we've really made commitments to these universities, Alabama A&M, CSUN, Cal Poly, Howard University as well, to not just introduce these novel concepts of space biology and planetary protection, but to really form relationships, because we know that's really what matters, and to be an integral part of their teaching process. So these are just a couple of slides. As Parag mentioned, he's there. Again, working in our labs here at JPL, and this was our first program called Micro, or Multi-Institutional Collaboration for Research Opportunities. I know we get very creative on our names as well, but this was the beginning. And then we did Euro, that took us out of the labs and actually put us in the classrooms directly with the students. And we had the opportunity to take this show on the road and work with the universities. And we worked with over about 100 kids at all of these universities. And so we really hats off to Region and Rachel and all the other schools that were part of this road tour. We really had a ball. And then really getting into, once COVID hit, we had to go virtual. And this is kind of where we are now with Voices. And this was virtual opportunities. And we wanted to continue this outreach. And we really created a platform that was successful. And once we figured out that Ames was doing the same thing, we said, wow, we need to really come together and really collaborate on this to reach more students. And so now with all of this great work, we've come together, and we've also had a 5-week internship opportunity there. But this has been a wonderful opportunity. We thank you so much to Parag and his team, Amanda, everyone, for being a part of this. Obviously, Lisa and Armin are integral on the JPL side for getting this done. And thank you. Enjoy this. I'll be around for a little bit, but I have to jump on and off. But thank you again, and enjoy this boot camp.
Thank you so much, Elvin. I would also now like to introduce you guys to our instructors throughout the boot camp, as well as some of our teaching assistants. So let's start with Lauren Sanders. Lauren, if you want to turn on your camera and mic and just introduce yourself.
Hi, everyone. Thank you so much for being here this morning. I'm really looking forward to this boot camp. My name is Lauren Sanders, and I'm a bioinformatician at NASA GeneLab. My background is in cancer genomics, and I've been taking those skills in the past couple of years and applying them to space biology. And I think, as you've heard from some of the other folks from space biology, we're really excited for you to potentially take the work that we've been doing here and teach it at your own institutions. And thank you so much again for being here.
Great, thanks, Lauren. You'll be hearing more from Lauren, especially tomorrow. Next, I'd like to introduce Lisa Guan. Lisa, if you want to introduce yourself.
Thanks, Amanda. Hi, I'm Lisa. I'm a scientist in the Planetary Protection Group at JPL. I've been a part of this program for 2 years now. This is my second year, and it was really a great experience last year. I'm glad to see some familiar names, and very much looking forward to the next couple weeks.
Great, thanks, Lisa. And next, I would like to introduce Mike Lee. Mike, if you want to turn on your camera and introduce yourself.
Hey all, my name is Mike Lee. I'm a biologist turned bioinformatician. I work with GeneLab as a bioinformatician now. My background's in microbial ecology and my expertise is really in metagenomics. And thanks all for being here. You'll already know, or you will find out, Lauren and Amanda are both amazing. They put together a ton of great information, so this is going to be great. Thanks a lot.
Thanks so much, Mike. Another one of our instructors, Armin Swalemanasian, is currently out sick today, but he'll be joining us later in the week, hopefully. So we'll have him come on and introduce himself the first day that he's here. With that, before we, and I'm Amanda Saravia Butler. I'm GeneLab's data processing lead. Currently, my background is in biochemistry and molecular biology. A big thank you. I know a lot of our managers and people that have made this possible and given us just a tremendous amount of support. Really big thank you to all of you guys. Could not be here without you. Also, a few of them put stuff in the link, or in the chat. David and Parag both gave links as well as their email address for you guys to learn more about the space biology as well as the Biosciences division. So please feel free to check that out and shoot them an email if you have any questions or want any additional information. So just want to make sure, just give one final opportunity for our managers if there's anything else you guys want to say before we dive into the content. Okay, thank you all so much for being here, and all of you guys have the link, so please feel free to hop in, hop out as you see fit, and as you have time, you're of course welcome to attend as much or as little of the boot camp as you'd like. Okay, so moving back on to our content now. Is there anyone who cannot see my screen? Okay. Don't see that. Okay, great. So hopefully that means you all can see my screen. So before we get back into the lecture here, just a couple of housekeeping stuff. I know that I have sent you guys a bunch of emails and links with all of this information before, but just to remind everyone, all of the content for this boot camp has been uploaded to GitHub, and so you can find it. I'll put this link in the chat here. All the content can be found on GitHub, including our schedule. So just to take a look at the bootcamp schedule, You'll notice that every day we have 2 20-minute breaks, one in the morning and one in the afternoon, as well as an hour for lunch. I am going to make sure that we stick to these timed breaks and lunches so that if you guys have anything that you have to tend to, or if there's someone that you have to meet with, I'll just ask that you try to schedule it during one of those breaks or lunches. I know at least a couple of you have mentioned that there might be a day or some sections that you'll be missing out on. That said, as I mentioned at the beginning, we are going to be recording this entire boot camp. As you go through the schedule, you'll notice that there is a mix of lecture and hands-on material as well as sections for debrief. So we are going to be going back and forth between lectures and our hands-on material through the SMCE system, which we'll all log into at the start of the afternoon session. And what you'll also notice is after each major section, we have these debrief sections. So this is where we're going to get really interactive and you're going to let us know how things are going, how that previous section was presented, and start to think about how you could incorporate that content into your home institutions. So the other thing I want to point out is you'll notice if you scroll all the way to the end, you'll see that there's nothing currently scheduled for the last day of the boot camp, which is Friday. Since this is the pilot for our educator boot camp, we really wanted to make sure we had enough time to discuss all of this very dense content that we're going to be going over in the next week and a half. And so just in case any session goes over, I'll update the schedule accordingly, but I'm always going to keep the breaks as scheduled, just in case you guys have things scheduled for that. So I might push this content down as we go, if we have to spend a little more time on a section or not. So we do have an entire buffer day available. So no worries, we won't have to rush through anything. We really want to make sure that you guys have a good understanding of all of this content and are going to be able to teach it. I believe that's all of our housekeeping until we start logging onto the system. So is there any questions about anything I just went over? Is there any of you who do not have access to the GitHub link or the Google Drive? You can put it in the chat. You can unmute your mic and let me know.
Quick question. So the Google Drive, which contains the lectures and the recordings, these lectures and recordings are also accessible through the GitHub as well. I just want to confirm that.
Correct.
Thank you.
Yeah, and just to note, as we go, I might make some subtle changes. Like for instance, I noticed in this introductory PowerPoint that the NASA video I had was from 2020, and they recently released a new one for 2022. And so I just over the weekend updated that video. So I'll put the exact PowerPoints that we go through up on both GitHub and the Google Drive as well. So everything should be pretty much what's there now, but just if there's any minor modifications that we make as we go. Any other questions? So I access Google Drive from a Gmail account rather than CSUN. Okay, so yeah, any of you guys who sent requests to me, I'll go through that during the first break and make sure that I grant anyone access who requested access. So does anyone else have any questions or have any issues getting access to the Google Drive or that GitHub link? Okay, great. So that says just going to our schedule here. We're in this welcome and then starting this lecture content. So we got about an hour before our first break. So I'm going to dive into this introductory slide deck here and we'll get started. If you guys have questions as I go, please feel free to type them into the chat. You can also feel free to unmute your mic and interrupt me. So I'm going to go ahead and get started first with an introduction to NASA and also to space biology. So this, we're going to start with the National Advisory Committee for Aeronautics, which came before NASA. So about 120 years ago now is when the Wright brothers first took flight in 1903. A little over a decade after that is when the National Advisory Committee for Aeronautics was established. And then shortly thereafter, we see the establishment of the first several centers, including Langley in 1917, the Ames Research Center, which is where we're coming from, established in 1940. In 1942 was the Lewis R. Glenn Center. In 1946 was the Dryden Armstrong Center. 1947 was the first time we had our very first supersonic flight. And in the 1950s is when this advisory committee begins to look into the possibility of manned spaceflight. And then in the late 1950s, they begin to plan an establishment to orbit a scientific satellite. And then, as many of you guys know, in 1957 was when the Soviet Union launched the very first satellite in space, Sputnik. And then shortly after that, the US followed suit and launched its first Earth satellite. At that same year, in 1958, was when NASA was established and absorbed itself into NACA. And then in the early 1960s is when we have Mercury's single astronaut program, begging the question, can humans survive in space? In the mid-1960s was when Project Gemini got started, in which 2 astronauts practiced space operations. And then, as you guys all know, in the late 1960s and early 1970s is when the Apollo project was running, in which we began exploring the moon. In 1981 was the Space Shuttle's first mission, STS-1. And then in 1984 was when Congress authorized NASA to begin building the space station. And actually 2011 was the last space shuttle mission. And since then we were actually relying on Russia to launch our astronauts into the space station. Until recently now we have commercial partners that can launch astronauts to the space station. And so NASA's vision is to reach for new heights and reveal the unknown for the benefit of humankind. And NASA's mission is to drive advances in science, technology, aeronautics, and space exploration to enhance knowledge, education, innovation, economic vitality, and to be stewards of Earth. And just here we have some images of some of the advances at NASA. Here on the left is the very infamous crawler, which transports rockets to and from the launch site at Kennedy Space Center. Down here you see astronaut Christina Koch doing some life sciences experiments on the ISS. This is just looking at some of the cell culture system hardware. We're going to get into that a little bit more a little later on today. And over here is just a future project of NASA, the Cygnus project, which is the Cyclone Global Navigation Satellite System, which is going to be helping with hurricane forecasting. So how does NASA achieve its mission? NASA headquarters, which sits at the top of NASA, provides overall guidance and direction to the agency. NASA centers and facilities are where all the day-to-day NASA work is performed in laboratories, airfields, wind tunnels, etc. And NASA conducts its work through 4 main organizations known as mission directorates. These mission directorates include the Aeronautics Research Mission Directorate, the Human Exploration and Operations Mission Directorate, the Science Mission Directorate, which is where GeneLab sits under, and the Space Technology Mission Directorate. And here's just the latest NASA org chart. I do have links on these slides for any of you guys if you guys want to learn more about any information that I'm presenting in this brief overview of NASA's history. I'll encourage you to do so. It's just how the overall organization of NASA looks. Our current leadership, so we are led by Administrator Bill Nelson and Deputy Administrator Pamela Malroy, and I have links to both of their awesome bios if you guys want to learn more about our NASA administrators. I encourage you to click on those links and learn more about the great background of our administrators. Here's a map of the U.S. depicting all of the NASA centers and facilities. As you can see, both GeneLab and JPL are out in California. So GeneLab sits in the Ames Research Center in Northern California, and then in Southern California is where the Jet Propulsion Laboratory is in Pasadena. And I do want to point out that we do have a robust history and a history team at NASA to describe everything that NASA has done in the past. And Parag actually put this link in the chat, as well as the JPL history link. So you can go to history.nasa.gov and learn more about the history behind each and every NASA center. So this is just a picture of Ames and JPL here and here. Since joining NASA, I often get the question of what has NASA done for us here on Earth? And so I have this slide up here just to highlight some of the advances that we use in our everyday life that is complements of the technology that's been acquired because of the research that has been done at NASA. So that includes things like satellite TV and medical imaging, ear thermometers, vision screening systems. The list really goes on, and of course, not to mention all of our advances in aeronautics. I'm often reminded by folks that work on aeronautics projects at NASA that aeronautics comes before space in NASA, so a lot of the advances in aeronautics we have NASA to thank for. So what's in store for NASA? A couple of vision of NASA's future is to extend and sustain human activities across the solar system, expand the scientific understanding of the Earth and the universe in which we all live, to create the innovative new space technologies for our exploration, science, and economic future, the advancement of aeronautics research for the benefit of all society, to enable program and institutional capabilities to conduct NASA's aeronautics and space activities. And then one of the reasons that we're all here and we get to do this is to share NASA with the public, educators, and students to provide opportunities to participate in the NASA mission, foster innovation, and contribute to a strong national economy. And so, of course, to do all this, we're going to need to do more science. So this is just drilling down now specifically into the Science Mission Directorate at NASA. This is just an overview of all the achievements of the Science Mission Directorate, including over 10,000 scientists funded, over 114 spaceflight missions, including several other technological developments and smaller missions that have been complements of the Science Mission Directorate. And again, there's a link here at the bottom if you want to learn more about the Science Mission Directorate. So the types of science that are done at NASA under SMD include Earth science, the study of our planet; planetary science, the study of the origin and history of the solar system and the potential for extraterrestrial life; astrophysics. What % of the budget is for education? I don't know that answer off the top of my head. Does anyone online who knows what % of the budget NASA is allocated for education?
So most of our programs have a little slice of education funding internally to that division that Amanda's referring to. So there isn't really a dedicated education office. There used to be many years ago. NASA's mission generally is to explore. Of course, education and training is part of that as we think about our pipelines and anticipate our technology needs. But organizationally speaking, there isn't an entire education office, if that helps answer the question.
Thank you, David. Yeah, good question. We also have under SMD heliophysics, which is the study of the sun and its effects on the solar system. There is the Parker Solar Probe, which is currently in progress, which I'm super curious about. It's going to get pretty close to the sun and teach us more about it. And also my favorite topic under the Science Mission Directorate, life sciences and life science research. So this is just a quick overview timeline of 50 plus decades or 50 plus years of NASA life sciences research. So we've been doing research in the life sciences from the biosatellite days back in the 60s all the way up through the shuttle missions in the 80s, 90s, and early 2000s. And then now we have a fully functional laboratory on the International Space Station where we conduct a lot of life sciences research. And then looking into the future, we're planning to do additional research as we move beyond low Earth orbit. So what has all of the past 50 years of life sciences research taught us? Well, it's taught us that exposure to spaceflight changes pretty much all of our physiological systems. So we see changes in the musculoskeletal system, the cardiovascular system, the lymphatic system, the central nervous system, even the reproductive system, as well as the digestive system. So it seems like the space environment does have an impact on our physiology as animals. But of course, animals are not the only ones. We also see an impact to plants as well. Here you're just looking at some images of plants grown here on Earth in those same types of plants grown in the space environment. You see the altered root structures both here and then on more the micro scale down below. And of course, microbes are also impacted as well. And because of that, we really want to learn more about how the space environment affects all of living terrestrial organisms. And the space biology program is a program that we focus on the life sciences research. So under the space biology program, we have research in animal biology, microbiology, cell and molecular biology, developmental, reproductive, and evolutionary biology, and of course, plant biology. And again, if you want to learn more about these programs, there's a link at the bottom of this slide. And I was also curious when I first joined NASA is how do we do all this life sciences research in space? Well, we do it with specialized hardware. So I just want to take some time. Got another question. Are there currently any morphological studies being conducted due to these aberrant forms of life? Yes. So a lot of the experiments in space are usually PI driven, the ones that are funded through NASA. And we also have a commercial side of the ISS, which funds research for commercial entities. And part of what they study is how all of life is affected, both on molecular level to the physiological level, including things like morphological changes in the various organisms that are studied. So this here is just looking at some of our hardware in order to do life sciences research in space. This is on the left here is the bioculture system, and on the right is a cell cult. So both of these systems are designed to allow cells to grow in space. So we could do cell culture work in space, and you'll see all the microfluidics within the hardware here to allow media changes as well as fixatives to go in so we can actually preserve the state of those cells in the space environment at various time points. Here we have the biological research in canisters hardware, more commonly referred to as BRIC, and this allows us to grow and study microorganisms in space. Here we have a hardware designed for smaller animals. So this is the multi-use variable gravity platform, more commonly referred to as MVP. And the cool thing about this is, well, you can grow small organisms such as fruit flies in here. You can bring them up in these chambers, release them into this chamber here, and that chamber is equipped with video surveillance as well as controlling the microenvironment there, recording the microenvironment. And the cool thing about this is those fruit flies can then go and lay their eggs, which can then be released into the next chamber, and so we can see entire life cycles and how they're affected by exposure to the space environment. The other cool thing about the MVP is, as the name implies, this is actually sitting on a centrifuge. So not only can we observe the effects in microgravity, but we can also, through centrifugation, allow different levels of gravity, exposure of different levels of gravity to the organisms in here and see how that affects their behaviors on both the macro and micro scales. We have another question here. Can cells be passaged in space? So good question, Alba. We can allow the cells, the previous slide here, one more previous slide. So we can allow the cells to grow in space by offering new media and media exchanges. Passaging is pretty difficult in the way that you're thinking in a standard laboratory here on Earth, so that tends to be a little challenging. And a lot of these devices, because crew time, our astronaut time on orbit is so precious, a lot of these are made to be self-sustaining, so very minimal interaction with the astronauts, unless, of course, they're taking them out. We do have microscopes on orbit, so we can take them out, and our astronauts will look under the microscopes, and that information is sent through telemetry to Earth, and so our scientists on the ground can have a look at what's going on in the cells in space. Let's see, I have a couple of other questions in the chat. Does all this strain on the physiology impact the immune system anyway? Yes, big impacts on the immune system. I heard the ISS is filled with microbes, so I don't know if that helps or harms the immunity of our astronauts. Would be cool to see a study of astronauts' immunity. Yes, I agree. So there is an impact on the immune system actually in most, if not all, of the model organisms that we've studied so far. The ISS is actually an enclosed environment, so anything that lives inside the ISS is because we brought it there when we go and we visit. And actually the astronauts go through a quarantine period before they actually go up into the ISS. And so if anything, the ISS is a lot cleaner and a lot fewer microorganisms than we're exposed to every day here on Earth. And so due to the changes that we do see in the immune system of all organisms, including astronauts, might be due to the fact that they're just not exposed to as many microorganisms in space. So we see a reduction in immune system functionality. One hypothesis is because there's limited amount of microbes in space. And then once they come back down to Earth, it's like a hyper exposure now to those microbes. Prague, thank you for putting in the chat more about funded studies. Trying to get through your questions, guys. Let's see. Yeah, so Elba, you can also grow cells in suspension in space with these cell culture equipment. Yeah, David Smith also mentioned a program that we have called the Microbial Tracking Project. So we do actually swab the International Space Station, and then we send those swabs down to Earth, and we perform things like metagenomics analysis to observe which organisms are currently residing on the ISS and how those organisms change over time. So a couple of our GeneLab data sets actually contain omics data from those microbial tracking projects. Yeah, and definitely, and I will be getting into some of the work from our analysis working groups, which I am going to encourage all of you to join. And we do, our analysis working groups do a lot of work studying the data sets on GeneLab and looking at things like immune function and how that changes in organisms flown in space. That's great, guys. So continuing on with some of the hardware, and feel free to continue to write things in the chat. I see a lot of our managers are putting in answers and helpful links. So guys, please feel free to check out those links. So I just went over the MVP platform for small animals. We also, of course, have hardware to grow plants in space. What you're looking at here is an advanced plant habitat, and over here on the left is our ground unit. So I should mention that you'll notice that a lot of these hardware look different than the hardware that we use to do similar studies here on Earth. And because of that, when we are performing experiments in space, any of the equipment, the specialized equipment that we use in space, we have equivalent ground-based units of those same habitats. So when we have our ground controls, they're housed in the same exact equipment just here on the ground to mimic any impact of that actual habitat and what it could have on the physiology. And so here on the left is the ground unit for the advanced plant habitat. And here on the right is that unit being used in space to grow a dwarf wheat. This is another habitat called the veggie habitat that we use to grow vegetables in space. And this is just looking at a series of images of the veggie habitat and some astronauts in space, taking a look at the lettuce that was grown in space and then enjoying some of that space-grown lettuce on orbit. And then the last piece of hardware I'm going to go over is the hardware we use for our rodent research missions. So here on the left, we have an animal access unit, which we position onto the habitat, which you see here on the right, in order for the astronauts to handle the animals without risk of them accidentally being released into the space station. And then down here on the lower right are just some images of what the habitat looks like on the inside. So this is capable of housing up to 5 mice on each side, so 10 mice total. And then this contraption here in the middle provides the water system via lixids to the animals. And we also supply food in the form of food bars, as you see here on the bottom right. And so one project that we have at NASA that brings together all of the studies that are done under the space biology program is NASA GeneLab. So before I dive into NASA GeneLab and open life sciences resources at NASA, I did want to share with you guys a little video just to conclude the NASA general overview. This is a video that NASA put out very recently talking about the future of NASA. So I'm going to go ahead and play this video for you all. Message me in the chat if you can't hear the sound. Okay, I hope you guys are all able to view that video, and on that slide is the link to that video in case you want to watch it again or share it with anyone. I provided the link on that slide. So that concludes our overview of NASA and space biology. Now I'm going to dive into GeneLab specifically and some of our other life sciences open resources that we have available. So our last decadal survey from the NRC was released in 2011. Our next one should be released any day now. And this, in the previous one, a little over 10 years ago, showed us the cost of sequencing has been drastically reducing ever since we mapped the entire human genome back in 2001. And this is just showing a comparison to Moore's Law. So it's been even faster than Moore's Law. So when this decadal survey came out, we are about right here for a cost of a raw megabytes of DNA sequence. GeneLab was started in 2014. And here is where we're at around today. So the cost has drastically reduced. And in that decadal survey, it was stated that genomics, transcriptomics, proteomics, and metabolomics offer an immense opportunity to understand the effects of spaceflight on biological systems, and that such techniques generate a lot of data that can be mined and analyzed for information by multiple researchers. And so to achieve that goal, GeneLab was created. So before we get into it, I know a lot of you are biologists and have strong biology backgrounds, but for the handful of you that don't, I'm just going to quickly go over what omics are. So in all of us, we are all composed of cells, and within those cells, we have DNA that is tightly and condensedly packed into chromosomes. That DNA contains instructions for life. And so back in the late 1950s is when Crick coined the term the central dogma of molecular biology. And back at that time, We knew that DNA could self-replicate. DNA can then be transcribed into RNA, and RNA can then be translated into proteins, and where proteins are expressed, which proteins are expressed, and when proteins are expressed make up life and everything that we see in our physiology. So initially we thought that this only took place in one direction. DNA makes RNA, makes protein. But of course, we now know that molecular biology is a lot more complicated than that. And so moving beyond the central dogma and studying different life forms, we now know that not only can DNA self-replicate, but there's also RNAs that can self-replicate. We know that DNA makes RNA, but RNA can also regulate and control DNA. We know that although RNA is translated into proteins, proteins can also regulate RNA expression, and proteins can also regulate DNA at the epigenetic level. And so to really understand what's going on at the molecular level in the space environment, we really have to understand what's happening for all of these different molecules. And so all these molecules together are referred to as omics. And specifically, anything looking at the DNA level makes up the genome, and anything regulating the genome would be considered the epigenome. At the RNA level, it's considered the transcriptome, and everything above the transcriptome would be the epitranscriptome. Anything involving proteins is known as the proteome. And then we have specialized molecules known as metabolites, which make up the metabolome, and this is just a breakdown of some of those metabolites. So why would we want to study omics in the context of spaceflight? Well, as I mentioned, the what, the when, and the where genes are expressed allow for cell type diversity. So this is what makes our eye cells different from our liver cells. It also enables us living organisms to be able to respond and adapt to our surroundings. And gene expression is primarily regulated by environmental factors, both at the micro level and at the macro level. So what you see here in this image on the right are all of the external stressors that come in the context of spaceflight, which can then in turn affect the molecular molecules in all living systems. And we know that spaceflight alters the transcriptional patterns and molecular signaling networks within our cells, which will then in turn cause physiological changes, as I went over in previous slides. So understanding such changes will enable development of mitigation strategies so we can better withstand the rigors of long-duration spaceflight. And I just want to point out here of these, what you see depicted here are the 5 major stressors of spaceflight. And if you look at the GeneLab repository, the majority of our data sets are looking into the effects specifically of radiation and changes in gravity fields. So speaking of GeneLab, GeneLab is the first multi-omics space-related database. We currently have over 370 publicly available studies, and these studies contain both raw data as well as process data for some of the data sets, and it also comes with environmental data associated with each study. And we have a visualization portal as well to view both the environmental data as well as the process data from these studies. GeneLab provides a variety of tools as well, including a user-friendly submission portal. So we encourage anyone doing a space-relevant experiment with omics to submit their omics data to be hosted on GeneLab. We also offer an analysis platform containing tools and workflows where users can go on and analyze their data. We have an abundance of tutorials and some online resources to be able to learn how to analyze and process data such as RNAseq. And we have a workspace available to allow our users to collaborate and share data and information in a private and secure manner. GeneLab has a sample processing laboratory, which is a state-of-the-art sequencing facility. So we have a NovaSeq instrument in there in which we're able to process and sequence samples in-house. And we also provide sequencing as a service for PIs. And GeneLab is made possible, and we rely a lot on our scientific community, primarily through our analysis working group, which I'll get into quite a bit in a few slides later. And our community allows us to create things like standard operating procedures on both the wet lab side and the dry lab side to make sure that we're staying as consistent as possible with the most up-to-date technologies. And if you go to our GeneLab landing page, you'll find yourself here, and I'm going to get into what some of these buttons take you to in a minute. But before that, some of the beauty of GeneLab, but life sciences open resources in general. For sequences, what methods do you use for low DNA yield and library prep? So we do have various kits and ranges of kits. There are some kits that are specific for low yield or really, really small samples that we can utilize. And actually, Lauren, maybe if you could put this in the chat, link to the GitHub for our wet lab team. So we have our standard operating procedures from our GeneLab sample processing laboratory team available on GitHub as well. So you can look a little bit more detail about the exact methods. We are going to go through an example of preparing an RNAseq study. But yeah, there's a variety of kits and stuff that we use to account for things like low yields. And library prep will depend on the question you're asking, which we're going to get into when we get into the RNAseq overview slide. Thank you, Lauren. So Lauren just put in the chat a link to GitHub, our sample processing GitHub page, which gives all the SOPs that our wet lab currently uses. So one of the great things about the open life sciences resources at NASA, including GeneLab, is that the goal here is to maximize knowledge by bringing all these experiments together in one place. And there's advantages to this. One is having smaller organisms. The smaller the organism is, the more samples you can have per payload. Space is very limited on orbit, and so working with smaller organisms when it's applicable or relevant to your research really helps maximize the N number that you can get. And of course, many of you guys being biologists, you already know the advantage of using some of these smaller organisms. A lot of them, their genes have several homologs to human genes, and so we can study some human pathways and stuff using these smaller organisms. A translational way. They are all identical, right? So you don't have to deal with a large amount of genetic diversity with these smaller organisms. They require fewer resources. They have a short life cycle, which means that you can actually study the entire life cycle in space, which is obviously something that you can't do with humans right now. And you can measure the response of the whole multicellular organism. And this is just an example of some things that fruit flies are used for to study here on Earth. And then, of course, as you get larger into animals, so moving more towards mammals and rodents specifically, we increase the relevance to humans. So the caveat being we have a smaller N number, but now we're getting closer to humans, closer to translational studies. We are, of course, more microbe than we are humans, so we have more microbial cells in us than we do human cells, and microbes are responsible for getting us sick as well as making sure that we function properly. So understanding microbes and how they're affected by the space environment is critically important. Similarly, we are never going to be able to survive anywhere where we can't have plants, so understanding how plants respond to the space environment is also very important for long-duration spaceflight. And then looking at all of these systems together is considered a multi-omics or systems biology approach. So having resources like GeneLab and other life sciences open resources, which I'll talk about in the next few slides, allows us to look at all living organisms as a collective system. And so this can really allow us to identify any shared processes or molecular signals that we're seeing affecting all terrestrial life in the space environment. So as mentioned previously, NASA has a variety of biological open science resources now, GeneLab being one of them. So this is just looking at 4 such resources. Just to quickly go over these, we have a biospecimen sharing program. So anytime a PI is selected to do a rodent research mission, the biospecimen sharing team is notified, and any organ or tissue that the PI is not needing further research is then used by the biospecimen sharing team and archived in the NASA Biological Institutional Scientific Collection, known as NBIS, to host preserved samples from spaceflight missions. And those are all findable and archived within the Ames Life Sciences Data Archive, known as ALSDA. And so within ALSDA, anyone can request a tissue that has been preserved from a previous mission that hasn't already been acquired by another PI. So you guys can go to ALSDA and look for a tissue that you might be interested in to see if one's available for from spaceflight mission. All that you have to do is just provide a justification for what you're going to be doing with that tissue and show that you have the funds, show that you have funding to be able to do the science that you propose to do with that tissue. Also, an ALSDA is a host to all data that isn't omics. So any non-omics data from spaceflight missions that are available is hosted on ALSDA. And I'm going to get into a little bit more about ALSDA a little later on. And then, of course, GeneLab is the place that you go to if you want any omics data. So GeneLab hosts all omics data from spaceflight and space-relevant missions. So just a quick overview of the GeneLab data systems. So GeneLab receives data from essentially three main mechanisms, one being GeneLab generated data. So as I mentioned, we have a sequencing facility in-house, and so we sequence tissues. We request them through ALSDA just like anybody else would, and when we have the funds for it, we will select tissues, work with our science team and our analysis working group members to identify tissues that would be of most interest, sequence them in-house, and then make those data available right after their sequence. We also have PI submitted data. So again, anyone, including any of you who's doing any space relevant research in which we will be generating omics data, we encourage you to submit your omics data to GeneLab through our submission portal. And I'll go over our submission portal in the next few slides. We also scrounge external databases. So other databases we look through, our users look through and try to identify studies, space relevant studies that would make sense to host on GeneLab. And when we do identify those studies, we ingest those data directly into GeneLab. So that way, the goal of GeneLab is to be a one-stop shop for anything space-relevant omics. And in addition to looking through external databases, we also look through publications as well to identify space-relevant data that we would want to then ingest into GeneLab. So all of those data sets go through our submission portal to then be hosted on our database. And our database is equipped with a federated search, so you can search data on GeneLab as well as you can search a variety of other databases as well through GeneLab. This is all hosted on the cloud, and we have a variety of applications to allow access to the data and make the data searchable and findable. And some of those applications are up here, and I'm going to go over some of these in a minute as well, but we have a data repository which hosts all of our data sets. We also have a workspace, what I had mentioned previously, to allow users to upload data and share data with collaborators. We have an environmental data portal to view environmental data associated with our studies, with the studies hosted on GeneLab, an analysis platform to actually analyze data that's powered through Galaxy, a visualization platform so you guys can visualize the process data through that portal. And we also have an API to make things more programmatically easier to push and pull or to pull data from GeneLab. And so all of our data, we follow FAIR principles, so findable, accessible, interoperable, and reusable. And I'm just going to dive in a little bit more to GeneLab and how to get to all of these great applications. So going back to the main landing page for GeneLab here, as you can see, if you go to genelab.nasa.gov, this is where you will end up. And so you can get to any of the applications I just mentioned by clicking on any one of these buttons. So first I'm going to quickly go over the Geode or Submit Data button here. And so the GeneLab environment for online data entry, which we refer to as Geode, allows users to upload or submit data to us, and they'll be requested to provide sample level data information as well as information about the assay that was used to generate the data that's being submitted. All the terms are connected to a controlled ontology to help them be more easily findable. And if you are a PI submitting your data to GeneLab, you will be provided with a study preview, so a private link so you can see what your data set will look like before it goes up and be able to review it and give feedback so we can make any changes before it goes public. And as I mentioned, space biology PIs as well as any PI who has space relevant data can submit data through Geode. This allows for efficient input of sample and assay level metadata alongside the data files that are being submitted. They're tailored to specialized needs of spaceflight samples. So you'll see a lot of these metadata categories are going to be pretty relevant to spaceflight missions. And we are now exploring extending this portal for use on ALSDA submissions, I'll get into a little later. So that's our submission portal. Next, I'm going to quickly go over our data repository, which you can get to by clicking the data repository button on our landing page. And when you click on that button, you'll be here in our repository. Our repository contains functions to search the repository, so you could type in keywords in this box here. The default is to search through GeneLab, but as I mentioned previously, we federate from other databases as well. So if you were to want to also search your key terms in these other databases listed here, you can just click on them and do that. We also offer a variety of search filters that you can play around with to pick and choose which data sets are going to be most relevant for you. And then once you do input your search criteria, you'll be notified of the number of pages and total studies that are consistent with your search criteria. And once you click on a study, here we see an example for GLVS 137. What you'll see here on the left is a navigation toolbar to help get you to the various sections of each study. So every study will have a study description describing what was done for that study. It'll have a protocol section providing all protocol details for what was done for the samples in the assay. We provide a sample table to show what's unique about each one of those samples and sample characteristics. We also provide an assay table to indicate information related to the assay that was used to generate the data being hosted. We also have a publication section, so any publications that were made using the data that are hosted in that data set, you can reach in our publication section. And then, of course, all of the files, so raw data files, and then if this is something that we've also processed in-house, the processed data files will be in that study files section. And then last but not least, for all processed data available, those processed data can be viewed through our visualization portal. I also just want to point out here towards the end of the description, if you guys are using one of these data sets in a publication, we have a link here for how to cite that data set in your publication. I also want to point out that if you click on the samples, the sample section of the toolbar, what you'll see is this sample table, as you see all the way down here on the bottom, containing sample level information. And you can also pick and choose which columns you want to see. And if you want to export these data, you can export it as a CSV file. So you basically click any column containing the information that you want from the study, and then you would click Export CSV, and it'll prompt you to save that CSV file on your local computer. And then just jumping down to the study files section here. So this is where all the data files can be acquired. So you'll notice here that if there is processed data, it would be in this GeneLab processed, in this case, RNAseq files. And then we publish processed data at various levels of the processing. So that way users at any level, depending on your experience, can come on and be able to take process data files maybe earlier on if you're a skilled bioinformatician and you want to process them yourself, or if you're not as well versed in bioinformatics and maybe you're just interested in something a little more downstream, like looking at the differential expression analysis, you can also grab that as well. So you can click on any one of these folders underneath. You would click on this little arrow to expand this section, and then once you click on one of these subdirectories here, those files will populate down here in this table. And it'll give you information about the size of the file, which resource category, which is equivalent to the level of processing that it is, and a short description about the file. And then again, you can select which files that you might want to download, and then you can click Download Selected Files, and those files will be downloaded to your local computer. So just a quick overview about what the repository currently contains. So we currently have just over 370 studies, which make up about 422 data sets. And of those data sets, we have 45 different species and more than 10 assays in total, and this all makes up over 135 terabytes of data currently on our repository. Just a little breakdown about those studies. So here's a breakdown based on study type. So just over half of the data sets you'll find are from spaceflight missions. So these are studies that were actually flown in space. We also have about 40% of the studies that are ground-based studies. So these are experiments that were done here on Earth to mimic some aspect of the space environment. And then we also have a small percentage here of high altitude studies. So these are studies that are done, let's say, in like a hot air balloon or parabolic missions, more commonly known as the vomit comet, where it experiences some time of some altered gravity state before returning to Earth. Here's a breakdown of the repository based on organisms. So you'll see that the majority of the organisms that we have are currently rodents, although we also have a good amount of microbes and plants as well. You'll see a piece of the pie here indicated as human. So we don't host any astronaut data on GeneLab. However, there are data sets such as bed rest studies, which mimic microgravity, that contain human data, and also human cell lines as well. And we also have other organisms, including fruit flies, invertebrates, fish, and then another small percentage here just of other organisms. Just looking at a breakdown, a word cloud here of the primary factors. So most is spaceflight. We also have quite a bit of radiation studies mimicking cosmic radiation exposure, altered gravity or hypergravity studies, as well as simulated gravity, and this down here is bed rest studies. This is a breakdown of our repository based on assay type. So this big chunk here of almost 70% of our studies are transcriptomic studies. And then if you look at those transcriptomic studies, we have almost 60% of those are microarray studies, and about 42% are RNAseq studies. Our RNAseq studies continue to increase. And RNAseq is, of course, what we are all here to go over during the boot camp. We also have a variety of other assay types on our repository, including protein expression, metabolites, metagenome, whole genome sequencing, genome-wide association studies, and some DNA methylation as well. And this is just looking at the growth of our studies over time. So this is number of studies in blue, number of data sets in orange here. And as you can see, we continue to grow and add to our repository and hope that that trend continues. So next, now that we've gone through the data repository, I'm going to quickly go over environmental data. So we also host environmental data associated with the studies. So this will include things like temperature, relative humidity, CO2, and radiation exposure. And you can also put these side by side and compare them with different data sets. So just looking at some of those environmental data over time. Here's a dosage of radiation. Here's temperature, humidity, and CO2 levels for emission here. So in addition to taking a look at environmental data, if you want to look at the omics, the processed omics data, we have a data visualization portal for omics data that have been processed. And this consists of things like principal component analysis plots, a query table where you can query differentially expressed genes, a heat map, and also a volcano plot. So throughout this course, we're actually going to be making static PCA plots, heat maps, and volcano plots. We are also going to be creating a static differential gene expression table. So we'll go over all of these in great detail during the boot camp. And I should say that through our visualization portal, all of these are interactive. So you could move these around. You could zoom in to certain locations on the heat map. You can query your favorite gene in the query table here, and you could make modifications to the volcano plot to change the p-values and different things that you want to look at. And the whole goal of this and providing all of this different levels of data is to democratize those data. So as you guys know, that when you have raw sequence data, raw omics data, it's very difficult to interpret and understand that unless you are a super specialized biologist known as a bioinformatist that knows how to handle those data. And so because of that, we work with our scientific community to develop consensus pipelines to be able to process those data to make it more accessible to more scientists. So once we change those raw data and process it to the level of differential expression. Now that data is a little more accessible to more scientists. And then when we, through our visualization portal, show pathway changes in these different data sets, now even more folks can understand and interpret changes in pathways. And then equating those pathway changes to potential changes in disease states, we can now involve more of the community to look at and review these data, reuse these data, come up with new hypotheses with the ultimate goal of really understanding what's happening to our terrestrial biology in the space environment. And so far, I know that these next few slides are going to be pretty busy. I just want to highlight all of the awesome work and publications that have come from using GeneLab data. So the reuse of data enables new discoveries. So here's just a snapshot of some of the 40 enabled publications thanks to GeneLab data. 10 publications have been produced by our analysis working group members. And so you guys have these slides, and so I just wanted to highlight some of this work and the awesome science that has come of it. Things like answering your question about what happens to our immunity in space. Some of that work is involved in these published papers. And so a lot of these papers, as I've been alluding to a few times, are complements of GeneLab's analysis working groups. So our analysis working groups consist of over 250 scientists from multiple space agencies, international institution in industry, as well as academia. The scientists meet monthly with each of their groups to analyze data in the GeneLab repository and come up with new hypotheses and perform different types of downstream analyses to learn more about what's going on in the space environment from GeneLab data. Our AWGs are broken down into 4 subgroups. The animal subgroup, which focuses, of course, on animal organisms and the effects on animals in spaceflight. We have a plants group, which focuses specifically on plants. We have a microbes group, which looks at microbes and the effects of the space environment on microbes. And then we have multi-omics, which is currently our largest group, and they collectively look at everything together, all levels of omics data and all the different organisms, to try to understand how the space environment is affecting terrestrial biology. And we are completely open, so anyone can join in AWG. You can participate as little or as much as you have the time to. I highly encourage all of you guys to join our AWG. So up here is a link for information for how to join in AWG. So I really encourage all of you guys to go in that link and become a member. This is just a quick screenshot of some of our members through our analysis working group that have been driving data reuse. A lot of these folks were involved in a paper package, a cell paper package that was put together a couple years ago, and were authors on some of the papers that came from our AWGs. So just to give you more links for different research that has happened thanks to our AWGs in providing data on GeneLab, here's a link to those 10 papers that the AWGs have published since 2018. So I encourage all of you guys to click on the links and read more about the awesome discoveries that have been made using GeneLab data. And then just to spam you a little bit more, some more highlighted works from our AWG members. And this is papers that came from the cell bundle package that I had alluded to. So definitely encourage all of you guys to take a look at some of the awesome research and new discoveries that have been made by reusing GeneLab data. And then kind of lastly for GeneLab, and I want to just talk a little bit about ALSDA. Before we get into the break, is GeneLab is able to accomplish everything that we are through 5 different sub-teams that all work together. So we have a science subgroup, a sample processing lab, data systems, which maintains all of our backend and makes sure that files are accessible and easy to download. Our data processing team, which I currently lead, and we're responsible for working with our scientific community to come up with standard processing pipelines and to process all of the data that we ingest and host on GeneLab. And then our science communication team to get the word out. And so all together, we come up with standard SOPs, both on the wet lab and on the dry lab side, and make all of our data and all of our SOPs publicly available in hopes to generate new knowledge by our users coming on and utilizing the data that we host. And I just have a couple more slides that's talking about integrating data beyond omics. And in 2021, so just last year, the Ames Life Science Data Archive transitioned to start using the GeneLab data systems. And the goal of this integration was to develop a multi-project submission portal to reduce the burden on PIs submitting data and enabling discovery of both omics and phenotypic data. So again, any non-omics data is hosted on GeneLab or hosted on ALSDA. So you see some examples here of some spaceflight missions that have been conducted, and those data are available on ALSDA. So anything about hardware, some environmental telemetry, raw and processed non-omics data is going to be on ALSDA. And the goal of this, of course, is to allow users to reuse these data to come up with new discoveries. And ALSDA transitioned their legacy database to a new infrastructure to comply with FAIR and open data. So they're currently working on complying with FAIR principles. One of the first steps in doing this is to establish consensus on standards for the assay sample and study design metadata, which are collected from PIs for their study, to ensure that those data can be reused. So to make sure that there's enough metadata associated with the study data in the repository in order for users to come on, understand the context of the experiment, to be able to interpret the data that's hosted. And since we are in the earlier stages of establishing those consensus standards, the ALSDA has established an ALSDA analysis working group. So there's currently about 100 members. They have monthly meetings as well to, again, come up with terminology and consensus on what standards to use in their repository. So I have an email address listed here on this slide. If any of you guys are interested in contributing to these standards, I encourage you to email that email address and let them know that you're interested in joining the ALSDA Analysis Working Group. And again, the whole goal, just to bring this back together, is to have multiple databases to form an entire knowledge-based system. So allowing us to take in data, to process those data, both non-omics and omics data types, and bring them all together to start understanding what happens in the space environment. And then our next step is to take that one step further and be able to use those data and those metadata to come up with a way to predict what might happen to humans as we travel further into space and are in space for longer periods of time. So we recently have a machine learning AI project, which Lauren, who's one of the instructors for the Bootcamp, leads, and this is where they're building using AI and machine learning to kind of build these knowledge graphs to be able to start making predictive models about what's happening in the space environment. And of course, the goal of that would be to be a knowledge-based predictor of what's happening in the space environment, and we're hoping to establish a modeling hub coming this December. So with that, this brings us to our first break.